This last chapter is super interesting. This is all about development. So we're going to talk about things like pregnancy. We're going to talk about how the egg is fertilized, how it moves down into the uterus and implants, and then we'll also really get into, I don't know if I have one on here. We'll also really get into the stages of pregnancy, like what happens in your first trimester, the second, and the third. And then on Thursday, we'll talk more about things like going into labor and delivering, the stages of labor. We'll talk about that, okay? Nursing and breastfeeding. So the term pregnancy, this term basically just describes everything that takes place from the time the egg gets fertilized until the time that baby is born. That's all of that is considered pregnancy. There's a couple of terms that I'll use as we go through this chapter. The first one is called conceptus. This is the developing offspring. Gestation period. The gestation period is essentially 280 days. And remember, your cycle, day one, is the day you start your period. And so whenever we talk about the gestation period, we always start from the very first day of your last menstrual cycle. Okay, so if you find out you're pregnant, you pee on a stick, find out you're pregnant, you call your doctor. That's one of the very first questions they're going to ask is, what was the date of your last period? That way they can figure out what your due date will be, because it's 280 days from that. Okay, now, this number, this little, what is that called? Tilde? I don't know. It means about 280 days. You know, it is not an exact science. They're going to tell you, what is yours, December 21st or something like that? The 20th, yeah. So they might tell you it's the 20th, and you might go on the 15th, or you might go later than that. It's really rare that you go the day they say. They have a little wheel that they'll calculate out what your due date will be, but honestly, like that's, yeah, it doesn't matter. Baby's gonna come when the baby's ready. Okay, so usually it's about 280 days. 40 weeks. Okay, I'll write that down. 40 weeks. A lot of times, doctors, they don't do it in days, they do it in weeks. So they'll say, oh, you're 12 weeks pregnant, or you're 39 weeks pregnant. So we do it by weeks. The embryo, that term is basically the offspring from the time it's fertilized up until week 8. So we always talk about it as an embryo. It's the embryonic stage. After week 8, all of the organ systems have formed. So beyond week 8, it now becomes the fetus. Okay, so from week 9 through birth, we talk about it as a fetus. Okay, but before then, it's the embryo. So this is just showing you sort of the progress from fertilization. You can see how tiny that is. Through the very first week, you can see this is a 3-week embryo here. At 5-week embryo, it looks like a little gummy bear. This is an 8-week embryo, and from that point on, we now consider it a fetus. And then this is a 12-week fetus. You can see how very similar it is. It looks just like a little baby. Everything has formed at that point. Really, all that that baby is doing is just beefing up in size from then on. So I'm basically going to walk you through all of those stages, from fertilization all the way up until birth in this chapter. So we have to start with the eggs, right? When you ovulate, you release an egg. One thing that's important for you to know is how long the egg is considered viable, meaning how long, once that egg gets released, what's the time window for it to be fertilized? It's actually really short. It's anywhere from about 12 to 24 hours. I would say 24 hours is on the long end. So usually it's within about 12 hours. So you have a really short window there to get pregnant. After about 24 hours, what happens is the outside of the egg, and you can think in your brain, think of like a chicken egg, right? The outside's kind of hard. The outside of an egg, when you release it, is also kind of hard. And after 24 hours, it gets even harder, and the sperm just can't penetrate it at that point. So it's only viable for sort of a short time. And then something else that's interesting is that sperm are actually viable in the female reproductive tract for up to 2 days. So, and some doctors even say 3 days. So sperm can live anywhere from 24 to 48 hours after ejaculation within the female reproductive tract. And that's kind of important because otherwise, if you think about it, it'd be nearly impossible for anyone to ever get pregnant. There would be no accidents if you ovulate and you only have 12-hour window for sperm to fertilize that egg. But thankfully, once sperm are released, they can survive in the reproductive tract for 2 or 3 days. So there might still be sperm in there when you ovulate. And ideally, what you want is as soon as you ovulate that egg, you want the sperm to be there. You don't want it to take a while for the sperm to get there. You want them already there. When the sperm fertilizes the egg, it means that the chromosomes from the sperm are combining with the chromosomes of the oocyte. And they form that fertilized egg, which we call a zygote. And so in order for the egg to get fertilized, we say that you have to have intercourse 2 days before ovulation, because remember the sperm can survive for up to 2 days. And usually we say 24 hours after ovulation is also best. Okay? So you want the sperm to be there. Now, one of the things that I mentioned to you in the last chapter is that typical ejaculate has about 250 million sperm in it. That's huge, right? And the reason is because most of those sperm don't ever make it out to the fallopian tubes. It's only 5 inches. It's only 5 inches from where the sperm get deposited in the vagina to getting out to the eggs in the fallopian tubes. But the reason why most of them don't make it out there is because a lot of that semen will leak out of the vagina. I remember doing fertility treatments, and before we really started trying, after intercourse, I would get up and go to the bathroom right away because I didn't want to get a urinary tract infection, and it would just all leak out. And so the first thing the doctor said was, "Don't get up. Just lay there. Prop your feet up on the headboard and just lay there. Kind of tilt your pelvis a little bit and let it be in there so that the sperm can make it out to the fallopian tubes." The other reason why a lot of those sperm get lost is because a lot of them are destroyed. Remember, vaginal secretions are really acidic. The sperm don't like that. And so the vagina itself will actually destroy some of those sperm. A lot of the sperm get lost. They get lost trying to make it up through the hole, the os of the cervix, or they'll get lost in the folds of the uterus. You also, ladies, have phagocytic cells in the uterus that are helping to keep foreign organisms, keeping infections at bay. And sperm are considered a foreign organism, right? They're not part of your own body, and so phagocytic cells will actually gobble them up as well. Usually we say only a few sperm will ever even make it to the fallopian tubes, and by a few, that means usually about 100. Sometimes it might be up to 1,000, okay? And that's just at the entrance of the fallopian tube. This is a picture, and I don't think this one is from your book. This is from another textbook. But this one's pretty interesting. So you can see the vagina here. You can see the cervix. Here's the os of the cervix. Here's the uterus and the fallopian tubes. So ideally what you want is you want the egg to be fertilized in the upper third of the fallopian tube. So you need the sperm out here. So this is showing you if you have 250 million sperm that get released here, that's 100% being released here. Okay. Only 3% will ever even make it through the os of the cervix. So 3% is about 12 million. That's still a lot. And then only about 0.1% will ever even make it up into the uterus. So we're now down to about 400,000. And then even up to the top of the uterus, only 0.001% will make it up here. So now we're down to 4,000. So the numbers that will even make it into the fallopian tube and find that egg drop significantly. So this is why we get down to only about 100. So this is why if a man has a low sperm count, this is why it can be really difficult, and a man can have fertility issues, right? The sperm count needs to be nice and high. The sperm have to become motile. They have to be able to move and swim and find that egg. And the other thing is sperm, we say, have to be capacitated before they can penetrate that oocyte. And so one thing that has to happen is they have to become motile. And motility of a sperm, not just that they have to be able to move and have a flagellum, but their movement has to be enhanced. So they have to be able to swim really well. And also the membrane of the egg has to become less hard. So that oocyte has sort of this hard covering, and it has to become less hard. And I'll talk about how that happens. The female reproductive tract will actually weaken the membrane that sits on top, the kind of the cap. We call this the acrosome that's on the tip of the sperm. And this makes sure that the enzymes don't self-digest the male reproductive tract. So really what's happening is when the sperm get released, those female secretions, those acidic secretions, sort of activate the enzymes on the head of the sperm. And it's those enzymes that help it penetrate through the egg. And it makes the coating of the egg a little bit weaker and softer. And that's really important. But you don't want those enzymes to be active when the sperm are still in the epididymis or moving through the vas deferens of the male reproductive tract. So they're not really turned on until they hit the female reproductive tract. It's the acidic secretions that turn that on. Something that's kind of interesting is that sperm follow an olfactory trail. What does that mean? What's olfaction or olfactory? Smelling. So weirdly, oddly enough, sperm actually can smell the egg. So they follow a scent trail to find that oocyte. So I mentioned to you that, if you think about the oocyte when it's released, think of it as like a chicken egg. It's really hard on the outside. And so the sperm have to be able to penetrate through that hard covering. And there's two coverings. One is called the corona radiata. The other is called the zona pellucida. So the corona radiata is on the very outside. And I think of the term radiata, like radiating out. So this is a bunch of cells on the very outside. So the first thing that has to happen is the sperm have to be able to weave through the corona radiata. Then they get to that really hard shell called the zona pellucida. And this is when those acrosomal enzymes really become active. They go through something called an acrosomal reaction. Those enzymes in the acrosome, that's kind of the cap on the head of the sperm, those enzymes will actually digest holes in that hard covering of the oocyte. They'll digest holes through the zona pellucida. You need more than one sperm to do this. So the enzymes that are found in the acrosome of one sperm are not enough to make that zona pellucida soft and to put enough holes in it for a sperm to get through. You need hundreds of sperm attaching to that egg, releasing the enzymes, softening the zona pellucida for one sperm to get in. And so, you've heard the term, the early bird gets the worm. That is not the case when it comes to sperm. So the very first sperm that hits that egg, all it's doing is softening the zona pellucida. And it's usually the one in the back. The very last one to hit that egg is the one that's now lucky enough to get in and make its way in. Now, only one sperm is actually allowed to penetrate. We call this monospermy, and we're going to talk about how monospermy happens. So what happens to ensure that only one sperm gets in? Why would you not want more than one sperm getting in the egg? What would be a problem with that? Think genetic material. Yeah, too many chromosomes. So usually if more than one sperm is able to fertilize that egg, usually there's going to be issues as it's dividing and it won't even implant. If it does implant, usually you'll have a miscarriage pretty quickly. Your body knows that it's not right, that it won't be able to develop. So this is a picture that's showing you this acrosomal reaction, right? So over here, you can see here's the egg. Okay. You can see the zona pellucida is kind of a gray line. So that would be, think hard chicken egg, right? The shell of that chicken egg right here. And then the corona radiata are these cells that are radiating out. So I told you that first the sperm have to weave through this corona radiata. You can see all these sperm out here coming at this egg. Now you can see up close, they've just made it really big right there. You can see these sperm coming in, weaving through the corona radiata. Each sperm, the acrosomes, those enzymes in the acrosome, help to soften up this zona pellucida. And then eventually, it gets soft enough that one sperm is able to wiggle its way in. Okay? And once that one sperm wiggles its way in, there's some other things that are going to happen. So once one sperm has entered the oocyte, calcium levels inside that egg go up. Now, this is the end of anatomy, right? We have talked about calcium in AP1. We talked about it in AP2. Calcium is so important. We need it for so many physiological functions, right? We need it for muscle physiology, for muscles to contract. It's the go signal for a muscle contraction. We need it for cells to divide. We need it for glands to secrete. We need it for everything. It is not a surprise to me that this plays a huge role in fertilization as well. Okay, so as soon as one sperm gets in, calcium levels go up. And anytime calcium levels change in a cell, something's going to happen. So when calcium levels go up, that's going to trigger the release of zonal inhibiting proteins. We call those ZIPs for short. These are little vesicles found in the membrane. They sit right under the plasma membrane of that egg. And what happens is, as soon as those ZIPs get released, it causes that zona pellucida to harden back up. Okay, so all those acrosomal enzymes made that zona pellucida, which started hard, made it soft. It got little holes in it. And one sperm made it in, calcium levels go up, and that zona pellucida gets real hard again. Okay, so no more sperm can get in. The other thing that ZIPs do is that if there's any other sperm that have reached the egg, they automatically will detach. This is just making sure that you don't get extra chromosomes in the egg. Once you've gotten one sperm in, you've got 23 chromosomes from the egg and 23 from the sperm. You don't need any more. Right? So this one is just trying to show you that one sperm that has made it in. Here's the nucleus of the sperm down at the very bottom. That's where the chromosomes are, right? So our 23 chromosomes are in there. And you can see that that zona pellucida got nice and hard again, and this sperm is getting detached. That nucleus of the sperm, it detaches from the flagellum, okay? And the nucleus is going to move towards the nucleus of the oocyte. So they kind of get close to each other. And it's going to swell up and form something called the male pronucleus. And the nucleus of the egg is going to swell and turn into something called the female pronucleus. Fertilization happens when the membranes of the male and the female pronucleus break down and the genetic material, those chromosomes, combine together. And you end up with 46 chromosomes. So this is just trying to show you here. You can see the male, the head of the sperm, and you can see it turns into that male pronucleus. Here is the female pronucleus. You can see that the membranes around those break down and the genetic material combines, right? And we end up with a single cell that has 46 chromosomes, right? Half from mom, half from dad. This is now called a zygote. This is interesting. This is a microscope image of, you can see the oocyte here, and you can see the male and the female pronucleus right before the membranes have broken down and the genetic material has combined. It's pretty fascinating. So again, this is now called a zygote. And usually this is taking place during the very first mitotic division. So if we go back, I'm not going to go all the way back through mitosis, but y'all can see here, right, you've got these centrioles, you've got these spindle fibers shooting out. That should sound familiar. This looks like mitosis is happening, right? So this is that very first mitotic division. Now, that zygote, in order for it to make a baby, that one single cell that now has a full complement of chromosomes has to undergo billions of mitotic divisions. It's going to divide and make identical copies of itself, right? Because every cell in your body has the same genetic material in it, right? And so that's what's happening. That one single cell is now going to divide and go through mitosis billions of times. So I have a little video. I don't know if this will work. We'll see. Let's see if I can search for one. I haven't watched any of these, so I hate to play these. Okay. We'll wait. I'll see if I can find one later. Oops. PowerPoint didn't like that. Let's see. Okay. All right, so human development is kind of divided up into 3 stages. The first one is pre-embryonic. The pre-embryonic stage is basically from the time the egg is fertilized by the sperm, and it ends just after that embryo implants in the uterine lining. We consider that all pre-embryonic. Once it implants in the uterine lining, we consider it the embryonic phase. Okay, and this is what we're going to find is that zygote, as it goes through divisions, it forms those 3 germ layers, the primary germ layers, like the mesoderm, the endoderm, and the ectoderm. Does that sound familiar? Okay, it's going to form those, and that is when we mark it as the embryonic phase. And then the fetal phase, remember this starts around week 8. All the organ systems have formed, and so at this point it's just that fetus is just getting bigger. So we're going to go through kind of each one of those. We're going to start with the pre-embryonic development. So cleavage, that term cleavage just means divide. And remember, as soon as that egg is fertilized and the pronuclei membrane breaks down and the chromosomes combine together, we get 46 chromosomes, that zygote is going to go through mitosis. So as that zygote is moving towards the uterus, it's going through mitosis. So you're going to start with 1 cell in that fallopian tube, and then you're going to end up with 2, and then 4, and then 8, and it very rapidly keeps creating cells. The first cleavage happens at around 36 hours. This is when you get 2 daughter cells. So you start with that 1 cell with 46 chromosomes, it's going to cleave, and you end up with 2. Okay, so usually around 36 hours. At 72 hours, once you've hit that point, it starts going through mitosis fast. And at 72 hours, you have a ball of cells called a morula. Still has not implanted. This is still up in the fallopian tube. Eventually, fluid will start to accumulate within the morula, and it converts it into what's called a blastocyst. This has about 100 cells in it. This is what's going to implant in the uterine lining, usually about 4 or 5 days after ovulation. Okay? So when do we ovulate in our cycle, ladies? What day? If we have a 28-day cycle, what day do we ovulate? Right in the middle, right? Day 14. And so if you have unprotected intercourse around day 14, anywhere from day 12 to 15, right? You can fertilize the egg. And usually we say about 5 days later, that blastocyst is now implanting in the uterine lining. So this puts us at around day 19. So usually women around day 19, about a week after ovulation, if implantation's happening, they may get a little bit of bleeding. They may get something called implantation bleeding. Okay. So that blastocyst has some fluid in it, and it has a big clump of cells, like 100 or more cells in it. A big group of cells on the inside is called the inner cell mass. For short, sometimes we call it the ICM. This is going to eventually become the embryo. But then there's also a ring of very flat cells called the trophoblast. And this will become the embryonic portion of the placenta. And the placenta is what is supplying the nutrients and the oxygen and helping to remove waste from that developing embryo. And then that fluid that's in there is the amnion. It's going to turn into the amniotic fluid. So here's a picture that shows you all of this, right? So we can see that here is ovulation. So here is that oocyte just after being ovulated. And then you can see here's all those sperm kind of attacking that egg, right? Making it through the zona pellucida, making it nice and soft. One sperm is able to get in. Within about 36 hours, that first division happens. You can see we're still up in the fallopian tube. Within 72 hours, we have a big ball of cells called a morula. Okay, so we start with a zygote. Put that up here. And we have a morula. Okay, and you can see the morula here, still in the fallopian tube, but you can see it up here, big ball of cells. And then eventually it's going to get bigger. It's going to divide more. We're going to fill it with some fluid, and we can see that here. This is when it first enters into the uterus. And you can see it up here. You can see this part right here is the inner cell mass that is eventually going to form the embryo. This part down here is the trophoblast. So I can do this one over here. Here's the inner cell mass. Right? Here's the trophoblast. That's going to form the placenta. And then this is the amnion. This is the fluid. Okay, so this is going to form the baby. This blastocyst is going to implant in the uterine lining. So remember how in the last chapter I showed you the cycle, the 28-day menstrual cycle? And you can see that even after you ovulate, the lining of the uterus is still getting thicker and thicker and thicker. And the reason is you want it to be nice and thick and pillowy for that blastocyst to implant. And again, this is happening, this implantation is taking place usually about 5 days. Usually we typically say about a week, so 7 days. You can even see that up here. But this is implanting usually about a week after you've ovulated. Now the blastocyst can actually float within the uterine cavity for a couple of days before it implants. That's why we usually say about a week from ovulation to implantation. And that blastocyst, it still has a ton of cells in it, so it still needs nutrients, but it's getting nourished by the uterine secretions. Eventually it gets big enough that it's going to need a blood supply, though, so it has to implant in the uterine lining. Again, implantation happens about a week after ovulation. And it's through chemical signals that it's implanting in the uterine lining. You actually have some inflammatory responses that are taking place in the lining of the uterus, the endometrium, and it's causing, if we think about the inflammatory response, right? It gets red, it gets swollen. It's getting red and swollen and hot because you're getting more blood vessels to the area. So as soon as these chemicals are being released, the uterine lining is becoming really inflamed. We're getting a lot of blood vessels in there because it's getting ready to nourish an implanted embryo. If the endometrium, for whatever reason, is not ready, or if it's unhealthy, so if you have an IUD and the lining is not thick, or if you've taken Plan B and the lining is not ready or it's inhospitable, then implantation can't occur. And the egg will just come out along with you'll shed your lining as well. There are some genetic mutations that can also prevent implantation from occurring. So again, this would be like polyspermy, 2 sperm were able to get in. A lot of times that it won't even form a zygote and it won't implant. If it does and it implants, you usually don't carry that to term. So this is a good picture because it's showing you that zygote. So again, you can see the inner cell mass. You can see this trophoblast, which is going to form the placenta. Here is the uterine lining, and you can see all these blood vessels. So you can see that basically that zygote just sort of digests its way into the uterine lining. Now you can kind of see why women have implantation bleeding, right? You get a lot of blood vessels in here. as this digests through that uterine lining and kind of implants in there, it's really easy for you to bleed a little bit. Okay? And eventually, it digests its way all the way into that uterine lining and it gets sealed up. Okay? By the 12th day, that uterine lining will grow over the blastocyst and it isolates it from the uterine cavity, and that's when implantation is complete. This is also when about 12 days after you've ovulated, you're probably normally getting ready to start another period. So your body has to say, ooh, don't shed the lining, we need to keep it. So you have to prevent menstruation. How do we do that? What hormone do we release? Progesterone, right? This maintains the uterine lining. So you got to release some progesterone so that you don't shed that lining. And that's, remember, what the corpus luteum is doing. The corpus luteum is releasing progesterone. How the corpus luteum knows it needs to hang around is because of another hormone called the human chorionic gonadotropin, or HCG for short. That's how the corpus luteum knows that you're pregnant. So this is not a hormone the corpus luteum is releasing. This is a hormone that's actually getting released from that implanted embryo. So this is actually secreted by the trophoblast cells. Later it'll be released by the chorion. And it's going to tell the corpus luteum, hang around, keep making progesterone. We need to maintain that uterine lining. It's also going to help to promote the development of the placenta. And usually HCG levels rise pretty quickly. We say they double about every 3 days. So they'll rise usually until the end of the second month, and then they'll sort of drop off, because typically after the first trimester, the placenta takes over and it makes progesterone and estrogen. And so HCG levels rise in your first trimester, and then they start to fall because you don't need a corpus luteum anymore. The placenta is making the progesterone. So when we did fertility treatments, I told you that my progesterone levels were really low. I had a couple miscarriages and I had to take progesterone. And I did it with a fertility specialist. When you do fertility treatments, they monitor you very closely. I had an ultrasound every week when I was pregnant. And at week 10, I was released to my OBGYN. And I remember still on progesterone, and I go to my OBGYN, and she's like, everything looks good. You can come off your progesterone. And I was like, no, I can't. I'm not ready to do that. She's like, I promise you'll be fine. Your placenta's making progesterone now. And I was so afraid to come off of it because I was afraid that I still wouldn't have enough. But I was fine. I came off of it. I think I came off at like 12 weeks, and I was fine. So this is just showing you ovulation and fertilization. This is showing you basically blood levels of some of these hormones. So you can see estrogen and progesterone around the time you ovulate, they're still not super high. And human chorionic gonadotropin, once that zygote implants in the uterine lining, so again, about a week after ovulation, 1 week later, right, you can see that the levels drastically rise. Okay, so this is, that's not a week. This is not in days. A week would be here. There we go. So you can see about 1 week later, the levels of HCG have started to go up. You can see HCG continues to rise until you're at about 8 weeks. And then it'll start to drop off as the placenta takes over. And the placenta, you can see once it takes over, you can see progesterone levels really go up. So they get much higher. The other thing I want to mention about HCG, I'll go back and write it on here, is that this is also what you're testing when you take a urine test and test positive for pregnancy. So pregnancy tests look for levels of HCG in the urine, right? So when you pee on a little stick, that Clear Blue Easy or whatever, it is actually looking for levels of HCG. You should not have any unless you're pregnant. Now, in order for you to have it in your urine, you have to have it high enough in your bloodstream. So usually levels in your bloodstream are elevated after just 1 week after ovulation. Levels start to rise. 2 weeks after ovulation is when they're a lot higher. And that's typically when it would come out in your urine. So this is why if you take a urine pregnancy test too early, it may come back as a false negative. If implantation happens outside the uterus, and this is possible, we call that an ectopic pregnancy. Some people will call it a tubal pregnancy if it happens in the fallopian tube. This is when the embryo will implant in that fallopian tube, and even sometimes the embryo can implant outside of the reproductive tract altogether. So let me go back to like this picture back here. You can see how the ovary and the fallopian tube, they're not touching each other. There's sort of a space between them. And you can see that the egg is getting fertilized way at the end of the fallopian tube. So it's possible that as soon as you ovulate, there might be sperm out here that can fertilize that egg here. And if that egg does not get pulled into the fallopian tubes, if it gets pulled out, then it's outside the reproductive tract. And it cannot grow, it cannot develop into a living fetus, and that's also considered an ectopic pregnancy. But what's more common is when it's fertilized in the fallopian tube, that instead of making its way down into the uterus, it just implants somewhere in the lining of the fallopian tube. And the fallopian tube does not have smooth muscle in it. It is not prepared to support a developing fetus. So this can cause a lot of abdominal pain. It can cause severe bleeding. It can cause the fallopian tube to become ruptured, which can decrease your chances of getting pregnant in the future. So usually, ectopic pregnancies have to be removed. A lot of times you can take a medication that will cause you to shed all of that. If that doesn't work, they'll have to do it surgically. And you want to have it removed before it grows and bursts that fallopian tube. Now remember, once that embryo implants in the uterine lining, we're also forming that placenta. And the placenta is sort of a combination between embryonic tissue and maternal tissue. It's kind of a mix between mom and baby. The placenta is usually fully formed and it's functioning around the 3rd month of gestation. So this is into your 2nd trimester. And again, remember the placenta, it's there to provide nutrients to that developing fetus. It's taking in oxygen. It is removing waste like CO2. It's also performing endocrine functions. So it is supplying hormones like maternal hormones to that fetus. One of the things that we talked about, at least in my class, we talked about in AP1, something called cradle cap. Do y'all remember what cradle cap is? Did you talk about it? So cradle cap is when a baby is born, they get these sort of overactive oil glands in their scalp. Now I want you to think about oil glands. My child is 7. He does not have any overactive oil glands, right? He doesn't have an oily face. Usually your oil glands are not overactive until you hit puberty and you start releasing sex hormones. So in an infant, it's a little weird that they would have overactive oil glands. The reason is because all the mom's hormones are getting passed through the placenta into the baby. So that includes all those sex hormones. So this is why it is not uncommon for babies when they're born to have this overactive oil glands in the scalp and they get cradle cap, like these flakes on their scalp. Also, when babies are born, their genitals are really large when they're born, and that's from all of those sex hormones that are coming from the mom. So hormones can pass the placental barrier. The 2 parts of the placenta, the mom and the fetal part, the maternal part of the placenta is called the decidua basalis. This is the part that lies just under the embryo. And the fetal portion is called the chorionic villi, or chorionic villus. Now, you looked at this one in lab. This is going to supply the umbilical arteries and veins. Sometimes, if maybe you are advanced maternal age, maybe you have, I don't know, a family history of a genetic disease, and you want to make sure that your developing baby does not have it, they can do what's called chorionic villus sampling. It's where they go in and they'll actually take a portion of the chorionic villus out. Reason they're going to do that for genetic testing is because it is made from the fetus. So it's got the fetal DNA, not the mom DNA. So that's why they would sample the chorionic villus. So if we look at this picture, you can see obviously the developing embryo here. And then here you can see in red, that's going to be where the placenta is forming. The decidua basalis, so the maternal portion, is up here. The chorionic villus looks like these clover leaf structures, and those are there. Okay, so you can see they're two separate portions. Now you can see it a little bit better. So again, you can see this decidua basalis, so the maternal portion out here. And again, the chorionic villus, those little clover leaf sections in there. If the hormones that are coming from the placenta are not enough, then the pregnancy will get aborted. So if the placenta doesn't form, if you're not making enough hormones, you'll shed your uterine lining. And so usually what we find, though, is that throughout your pregnancy, blood levels of estrogen and progesterone should continue to rise. This is doing a lot of things. Early on in your pregnancy, you're going to feel really tired. You're probably going to have to pee all the time. But other than that, you're not going to feel much different. And then in your second trimester, we call that the honeymoon phase. You're going to feel great. Like you're happy. Like you feel great. And then when you hit your third trimester, your levels of estrogen and progesterone are really high. And it is preparing your body for all kinds of things. So you're going to notice a lot of changes. Like your breasts will get much larger. They're going to get ready for making milk and lactation. You'll notice that you may start, like your feet may get a little longer. Your nose will get a little wider. That's because you're also releasing things like relaxin. Relaxin is a hormone that relaxes the cartilage in your body, makes it softer. It's doing this because of your pubic symphysis. You've got cartilage here that needs to get softer so that it can open and spread and get ready to widen that birth canal. So there's a lot of changes that happen, especially in that third trimester, physically. Normally, the maternal and the embryonic blood do not mix. So normally, just during gestation, you're not going to have mixing of that. There are some placental barriers between the maternal placenta and the fetal placenta. There are membranes in the chorionic villus. And those embryonic capillaries are continuous capillaries. So there's a membrane around those. So maternal and fetal blood don't mix. The only time they mix, typically, is if you're having a miscarriage or during childbirth. They can mix because all those membranes rupture. Okay? But until then, there should not be a mixing. So this is just showing you again that chorionic villus here. So again, those capillaries have a continuous endothelium around them. And then there's also that barrier between the mom and the fetal placenta. And so as that embryo is developing, once it's implanted, it's developing, it's growing, eventually we go through this process called gastrulation. Gastrulation is when the 3 germ layers form. And this is when we move from the pre-embryonic phase into the embryonic phase. So this is going to mark the beginning of embryonic development. There are some membranes that might form outside of the embryo, like the amnion. We know the amniotic cavity, it's filled with amniotic fluid. This is basically a sac that's in there. And the reason why you have this amniotic cavity is because it is providing buoyancy and it's protecting the embryo. So it's a cushion in there. I remember when I first got pregnant, my parents have a lake house and they have a boat. And I think I got pregnant in July and we were visiting in like August. So I was really newly pregnant. And I remember going to the boat and I went to jump in and I slipped off of the swim platform and I fell onto it and then fell into the water. I was very graceful. And then I fell into the water and I was so panicked, like, oh my God, like I hope I didn't hurt anything. But in reality, like there's so much cushion in here not only with your own organs and fat tissue, but also the fact that that embryo is surrounded with amniotic fluid. It's also helping to maintain a constant temperature. Now, you know, when you're pregnant, you don't want to sit in a hot tub for an hour, but you can take a bath. It's okay. Like, you can take a warm bath. I wouldn't make it so hot that your skin turns red, but you can take a bath because the amniotic fluid is maintaining the temperature for that embryo and that fetus. It also allows that developing embryo to move. And once it turns into a fetus, it allows the fetus to move. You're going to feel that fluttering that you get when that baby starts moving around. Once they're big enough, you'll be able to feel it. And the freedom of movement is really important because it prevents the body parts from sticking together and fusing together. So my brother and sister-in-law, they have monozygotic, monochromatic, identical twins. And that means that they shared an amniotic sac. The danger of having those kind of twins is that because they're in the same sac, there's not a lot of space for them, and they don't have less movement, and this is when you get conjoined twins. So all conjoined twins are the kind that share an amniotic sac. Okay, so there's a big danger with that. Amniotic fluid, it's actually just formed from blood filtrate, so it's formed from maternal blood. But then later on in pregnancy, you'll notice, like, if you ever see an ultrasound of a baby, a lot of times they're like opening their mouth. We talked about that in the digestive system. They're drinking amniotic fluid, and then they urinate it back out. And so the amniotic fluid is now no longer just blood filtrate from the mom, it also includes urine from the fetus. It's a little weird to think about. Early on during embryonic development, there's a yolk sac. And the yolk sac is basically hanging from the ventral surface, so the front of the embryo. This is going to form part of the digestive tube. This is actually the source of the earliest blood cells and blood vessels in an embryo. So again, gastrulation is basically the formation of those 3 germ layers. So once that zygote implants in the uterine lining, that inner cell mass starts to differentiate and gastrulate into these 3 layers: the ectoderm, the endoderm, and the mesoderm. Again, those should look familiar. The ectoderm, this layer, these cells are going to give rise to your entire nervous system, so brain, spinal cord, all your peripheral nervous system, and also your epidermis, so your skin. The endoderm is going to give rise to the epithelial linings in the body, so the endothelium. So it's the linings and the glands, anything that's in the body. And the mesoderm is everything else. So this is going to include things like your muscles. That's going to come from mesoderm. Your connective tissues come from mesoderm. Okay. So remember when we talked about connective tissue way back in AP1, we talked about how all connective tissue comes from something called mesenchyme. Those are the cells in your mesoderm, right? That's that layer in there. So all your organs are going to come from these 3 basic layers. And when the organs start to develop, we call that organogenesis. Organ and genesis. Genesis is the beginning or the formation. Right? So this is the formation of your organs. So this is just kind of a nice graph because it's showing you those 3 layers, right? The ectoderm, the mesoderm, and the endoderm. Now I like this graph because it's showing you that the ectoderm is on one side, and it's showing the endoderm on the other, and it's showing this red mesoderm in the middle because that's actually how it is formed. The mesoderm is in the middle, kind of sandwiched in there. So I remember the M in mesoderm for middle. So this is the middle layer, the middle germ layer. And again, this is a nice graph because it's just showing you what is forming in the ectoderm. What does it give rise to? Again, your entire nervous system, brain, spinal cord, and peripheral nerves. Also your skin. You can see the endoderm is going to give rise to endothelium, so all the linings of like the mouth and the digestive tract. And then the mesoderm is everything else. So it's going to give rise to muscles, your kidneys, your connective tissues, a lot of your visceral organs. So this is kind of the same thing. It's just showing those 3 layers, what forms in each of those layers. Again, you need to remember the ectoderm is giving rise to the skin and your nervous system. The endoderm is basically the lining of all of your body parts. And then the mesoderm is basically muscles and everything else. Muscles and cartilage, bones, right, blood, those are all connective tissues. Right, connective tissue up here. So the embryo starts with these 3 layers, and those 3 germ layers look kind of like a flat plate. Looks like 3 sheets of paper kind of stacked on top of each other. So here's a picture of that. Right? You can see it looks kind of like 3 sheets of paper: endoderm, ectoderm, and mesoderm. The mesoderm is in red in the middle. And eventually what happens is they sort of fold and create this little tube. And that tube is what's going to give rise to your nervous system. That's the neural tube. So you can see that here as well. So you can see those 3 layers. Oops, I didn't mean to move that. That over there. So we can see those 3 layers there with that mesoderm in the middle. Okay. And so forming those 3 layers is going to set the stage for starting to make your organs. So we say gastrulation. Again, this is making those 3 layers. Endoderm, ectoderm, and mesoderm. Sets the stage for then forming the organs, which makes sense, right? Because different organs come from those 3 different layers. So it's setting the stage for organogenesis. One of the very first things that happens during organogenesis is the formation of the brain and spinal cord. So at the 8th week, all of your organ systems have formed. And that is when we say that's the end of the embryonic period, and at week 9 we move into the fetal period. And so again, one of the first things that starts forming, brain and spinal cord. Usually the brain and spinal cord have started to form usually by the time you miss your period. So you're considered 4 weeks pregnant at that point. Brain and spinal cord have started forming at that point. So it's incredibly fast. So at the end of the embryonic period, so at the end of week 8, bones have been forming, and they form that primary ossification center, so they start becoming hard. You start forming those bones. Skeletal muscles have formed, and they can contract. Now, your skeletal muscles at week 8, they're not under voluntary control, but they can contract. Kidneys are developing. The gonads have formed. Lungs, digestive organs, those have all formed. Heart and liver. And the heart usually will start beating at 3.5 weeks. So again, 3.5 weeks is before you've even missed a period. So this is super fast that this is happening. So this is showing you a 7-week embryo. So you can see that it's within that amniotic sac. You can see that it looks like basically on an ultrasound, it looks like a little gummy bear. It has arm buds, it has leg buds. You can see that everything is formed here. So 1 week later would be the end of the, this is still considered an embryo. 1 week later is the end of the embryonic phase, and we move into the fetal phase after that. You can also see how big that placenta is. And I don't know if y'all saw this in lab. Somewhere. But we have a placenta. Did y'all see that in lab? The placenta? Oh, somewhere. Big. I don't know where it went. Oh. Y'all can pass this around. that she delivered after she had her baby, right? So you actually deliver your baby, then you deliver the placenta. So you're not done after the baby comes out. But y'all can pass that around if you want, just so you can see how big it is. Right now, obviously you can see here, even at the embryonic phase, you can see that placenta is huge. So once we hit week 9, we're now into fetal development. And during fetal development, the organs are still sort of developing and growing and getting bigger. And the fetus is going to grow really quickly after week 9. And it's going to go through a lot of physical changes. One thing that we're going to notice is that the circulatory system, remember, in a fetus is different from in an adult. So there's a lot of bypasses around the lungs because a fetus is not breathing air, and bypasses around the liver. The liver is doesn't really have to do a lot in a fetus because the umbilical cord is carrying those nutrients into that fetus. They don't need to be processed by the liver. Remember, some of the shunts are things like the ductus venosus. This is a shunt that's going to shunt blood basically directly into the inferior vena cava, bypassing the liver. Another one is the foramen ovale. This is going to shunt that blood from the right atrium into the left atrium. We looked at this one when we talked about the heart. And the ductus arteriosus, it's going to shunt blood away from the lungs. It's going to shunt it directly into the aorta from that pulmonary artery. So those are some of the bypasses, just to review those. We've talked about those already. You can see them up here. So the ductus venosus, you can see here. You can see the foramen ovale, and you can also see the ductus arteriosus. So those are some different shunts in the fetal circulatory system. Once a baby is born, the circulatory system will move into the adult pattern. Foramen ovale is closed, ductus venosus and arteriosus will disappear. So we can get that blood out to the lungs and into that liver. So you can see here, those shunts are gone, right? We've closed over all those shunts. This is a fetus. This is a 3-month fetus, so well into that second trimester. You can see it looks just like a baby, just smaller. This is a 5-month fetus, running out of room at 5 months. By the time you get to 9 months, there is not much room in there at all for that baby. Now, there are some birth defects that can happen. These are structural defects, functional defects. Something that I think is kind of interesting is about 31%, that's high, 31%, that's a third of all successful fertilizations will end in a miscarriage. And really, miscarriages are your body's way of getting rid of defective embryos. It's an embryo that may have not the right number of chromosomes, too many chromosomes, not enough chromosomes. It's usually genetic defects. About 10 to 12% of all newborns will have birth defects. And that number is low considering the fact that your body typically will miscarry if that embryo has a genetic defect. Okay? If our body did not naturally miscarry when there was a genetic defect, the number of newborns with birth defects would be much higher. So what causes birth defects? Lots of things. And the problem is we don't know. Some things we know can cause birth defects. So when you're pregnant, you have to avoid certain medicines, for example. But other things, these would be chemical things. Other things might be biological agents or physical agents that you might come in contact with. So maybe like a virus that you get when you're pregnant, or maybe you come in contact with something else when you're pregnant that can lead to a birth defect. All of these are known as what's called a teratogen. So these are some known agents. Things like progesterone. I know that seems weird. You make progesterone when you're pregnant. If you don't have enough, you take progesterone support, but you don't want your progesterone to be super high because it can cause birth defects. Thalidomide. Y'all have heard of like thalidomide babies, right? Have y'all heard of that? Thalidomide was a drug that was actually given to women back in like the 40s and 50s, and it was something that helped with nausea, right? So now you take like Zofran. Thalidomide was like an anti-nausea medication that they would give pregnant women, and it ended up the women who took that, their babies had pretty radical birth defects, and so now this is one that they do not give you if you are pregnant. You cannot take that. Rubella, the German measles, this one also we know causes birth defects. Alcohol, you don't want to drink, and radiation. You want to avoid x-rays and any kind of radiation when you're pregnant. Things that are suspected agents can be anything from antibiotics, insulin. Now obviously if you're a type 1 diabetic, you have to take insulin. Drugs for tuberculosis, and then all of these things listed up here. Barbiturates and antihistamines and antacids and smoking, and there's so many different things. So, you know, insecticides and herbicides. You know, if you're pregnant, don't spray Roundup outside. You let your husband do that or let somebody else do that. There's so many things that you just want to avoid when you're pregnant. You just want to try to be as natural as possible and not take a lot of medicines unless you have to. The effect of a teratogen is dependent on 3 things that I have listed up here. The very first one, which is probably one of the most important, is the time at which you're exposed. This is going to determine which organ systems are affected. So remember, what's the first organ system that starts forming in an embryo? Brain and spinal cord. So if you are subjected to a teratogen, maybe a chemical, or you get sick, and it's really early in your pregnancy, the organ system that will most likely be affected will be the brain and spinal cord. So the brain and spinal cord are affected throughout your entire pregnancy, but they're the ones that can get affected the earliest because they're one of the earliest to form. The other one is the nature of the agent. Some chemicals can affect, they're sort of broad spectrum, and they can affect any organ system that's developing at the time, but some teratogens specifically target particular organ systems. Okay. Alcohol, for example, is broad spectrum. If you're drinking when you're pregnant, and maybe during that time the ears are developing, it can affect the ears of that developing fetus. Methylmercury is something that targets specifically the brain and spinal cord, right? So again, that's forming throughout your entire pregnancy. And then obviously the dose. The higher the dose that you're exposed to, the greater the effect is going to be. So a mom who drinks every day while she's pregnant, it obviously is going to have a greater effect than that mom who drinks when she doesn't even know she's pregnant yet. Maybe she goes out and has a glass of wine, she's probably okay. But if you're drinking every day when you're pregnant, it's going to have a pretty big effect. This is not from your book, this is from another book, but this is pretty interesting because it is showing you the different phases. So pre-embryonic, basically remember this is before those 3 germ layers form. This is basically when the egg's been fertilized, it's moving down into the uterus, and it's just implanting. Notice that nothing is affected here. There's no organ system that's affected during that time. One, because it's super early, but the other one is there is no maternal blood going to that developing embryo during that time. It hasn't implanted yet. So you could drink, you could do whatever you want, and there's not going to be any effect during that time, but it's really early and the window is really short. Once it's implanted and we move into the embryonic period, this is from about week 3 to week 7, you can see if it's in red, that organ system is affected the most during that time. So you can see the brain and the spinal cord. Those are the first to be affected, and they're affected from the most from about week 3 up to about week 6, okay, of pregnancy. The other one that's pretty early is the heart and the ears. Now notice with the brain and spinal cord, it is still a bar that's on there all the way through the time that you have that baby. So again, taking any kind of medication can still affect the brain and spinal cord. The ears are also developing for a really long time, and they can be affected for a really long time.